So with that being said, I'd like to introduce Ed Lein. Lein is a senior investigator at the Allen Institute for Brain Science and an affiliate professor at the Departments of Neurological Surgery and Laboratory Medicine and Pathology at the University of Washington. It joined the Allen Institute in 2004 and has provided scientific leadership for the creation of large-scale anatomical, cellular, and gene expression analysis of the adult and developing mammalian brain as a catalytic community resources, including the inaugural Allen Mouse Brain Atlas and a range of developmental and adult human and non-human primate brain analysis. He leads the Human Cell Types Department at Allen Institute, which aims to create comprehensive cell analysis of human and non-human primate brains, understand what is disrupted in Alzheimer's disease, and create tools for precision genetic targeting of brain cell types as transformative tools for basic neuroscience and gene therapy. He's also a mentor or a member of the Brain Initiative Cell Atlas Network, otherwise known as BICAN, where he leads the Human and Mammalian Brain Atlases grant, and is also a member of the organizing committee of the Human Cell Atlases and CIFAR Fellow. And with that, I'll let Ed do the introduction.
Okay, can you see my screen okay?
Yes, perfect.
Terrific. Thank you so much for the introduction. It's really a pleasure to have the chance to introduce the session today, to tell you about the creation of really foundational human and non-human primate brain cell atlases and tools associated with those. This work is really supported by the NIH Brain Initiative, and in particular, an effort called the Brain Initiative Cell Census Project. This is a really large-scale collaborative national initiative to try to create an understanding of the basic components that make up the nervous system. This has been through two phases. The initial phase was called the Brain Initiative Cell Census Network, or BICCN, which has now entered a new phase called the Brain Initiative Cell Atlas Network, or BICAN, or BICAN, as you'll hear it referred to today. And this has really moved from trying to develop the tools to be able to understand cellular diversity in the brain and apply them to understand the organization of a model organism, a mouse in particular, to now being able to scale this up to really understand more complex, larger brains, most notably the human, but also non-human primates that are used as model organisms for biomedical research. In many ways, the aspiration of this effort is to understand the fundamental components that make up the nervous system. In spirit, this is really like trying to understand the elements in chemistry or the genes in the genome. For the brain, the basic components are the cells, and this can be abstracted into types of cells that collectively make up the brain and form functional circuitry that underlies all of our behaviors. And so an effort to try to get this foundational understanding is really an atlasing effort to try to characterize all the basic components, but also to understand their relationship in the functional organization of the brain. And so there's both a sort of a typology, as well as trying to put this into the three-dimensional coordinate framework of the brain, which you could think of as akin to a Google Earth, for example. What has made this really possible is a set of new technologies that allow one to measure the genes being actively used in individual cells, and to scale this up to be able to assay now millions of cells. And this allows you to take very complex nervous system tissues, to dissociate the cells or the nuclei of those cells, and measure all of the genes being actively used in those cells. That's a huge amount of information now to begin to cluster this data and to try to understand groups of cells that have similar molecular profiles that could be a cell type. And this can be really applied at scale. The tools of genomics have come to the rescue of the complexity of the nervous system to be able to systematically decompose the nervous system into its components. But unlike the history of the field, where one might have defined cells by the cell's anatomy or their connections, we now define the cells by the genes that they use. This creates a framework now a molecular taxonomy of types of cells that can be used then to begin to layer on additional pieces of information. So for example, we can understand the anatomy of a molecularly defined cell type, or the physiological characteristics, or their connectivity. And it leads us also to be able to not only describe, but to manipulate those cells in the future. So it's a very powerful framework that both characterizes and describes the cell's properties. A few years ago, the BICAN project had a really systematic effort to look at one part of the brain, the primary motor cortex, and really see whether this could work. If you brought together these various different ways of looking at the cellular organization of the brain, that you could create a robust classification of types of cells. And it turned out that this was extremely successful. In the primary motor cortex, for example, it was possible to define over 100 types of cells that could be described as a hierarchy from a high-level classes to an intermediate level of subclasses and finer types of cells, where we can get both the types of cells, the genes that they use, the state of their chromatin in the nucleus, the relative proportions of the cells, the spatial organization of these cells, and an increasingly rich understanding of the characteristics of these cells. And so this is really the epitome of what a brain cell atlas comprises. It is a robust description of the types of cells and a description of the properties of those cells that will eventually then lead to an understanding of the functional circuitry that the cells form for the brain as a whole. And one other key thing that came from this work is that it's possible to use this quantitative gene expression information to align cell types across species. These methods can be used in any species. They can be used in a human or a monkey or a mouse or any other organism for that matter. And in each case, one can describe the cellular diversity, but then you can quantitatively map across species in a way that's not been possible before to really align homologous cell types across species or to understand where there are differences. And this is a really key element because the human brain is very limited in the things that we can study. But we have experimental access in model organisms. And so we can gain a lot of information from studying model organisms that can be used to infer properties in human if we can align across species. And so this is a key element now to the future of these efforts where they are comparative in nature. We're trying to compare across species so that we can infer things up to the human, but also so we can understand where a model organism may or may not be a good may not recapitulate features of human that can be studied in the context of disease, for example. Just last year, a major publication package came out that really highlighted the power of these techniques and really sort of opened up a whole new field of what can be done now, moving across species, not limited to certain model organisms. And you'll hear about some of this today. It's really remarkably led to the first draft of an entire human brain cell atlas, as well as similar efforts in non-human primate, also efforts in development, in comparing across species, in understanding variation across individuals, and in characterizing the properties of these cells. So this is really a kind of a landmark achievement of the power of these techniques to be able to begin to characterize very large, complex brains with these methods. Now, this was really the pilot phase or the draft phase of this, however, and we've now entered the next phase of this effort called the Brain-Understood Cell Atlas Network, as I mentioned at the beginning, where the goal is really to try to make a comprehensive map or atlas of the human brain. And this creates some significant challenges that you'll hear about today. The brain is extremely large. It's extremely complex. It's not very accessible to study, and there's a lot of variation between individuals. And just in case you don't notice here, up in the upper right of this picture is the mouse brain, where it's been possible to get a very comprehensive map versus the human brain, which is much larger. So this creates some significant challenges. The project that you'll hear parts about today are part of a project called the Human and Mammalian Brain Atlas, or HMBA project, that really tries to bring together the collective skills and expertise of this community to be able to tackle this challenge of making brain-wide atlases of the human, macaque, and marmoset brain with different data types or different data modalities, and really also try to link this into a place where it can bridge across fields. So move from neuroimaging that can be done non-invasively to this cellular and molecular level, and to create a community resource out of this that can hopefully really accelerate progress across the whole community. On that note, I also want to mention that this is not meant to be something that we just create publications on. This is meant to be a transformative resource for the community, similar to the human genome, where people can map their data to this and add their information to this to create an increasingly rich knowledge base that the community can tap into and start to speak the same language and move forward to understanding what this means. This includes a whole variety of tools, trying to make formal versions of these cell atlases or references, to impose formal nomenclatures on these, to create tools to visualize this. One in particular you'll hear about today is the ABC Atlas, to be able to visualize these data, and tools to map your own data against this reference and transfer the labels onto your own information, as well as databases to aggregate information about these cell types. And so really this is meant to be a community resource at the end of the day. I want to close on one final point, that we really firmly believe in diversity in science. We believe that that fostering diversity produces much more equitable and impactful science. From the NIH perspective, this is referred to as the Plan for Enhancing Diverse Perspectives, or PEDP. In practice, this turns into many opportunities for training and mentoring, for outreach and community events, for support on the use of these tools. The link here provides access to a number of these efforts that are being led by our team here at the Allen Institute. And so with that, I'd like to close. I'd just like to really acknowledge the amazing group of people that are contributing to this. As I mentioned earlier, these are major consortium efforts. They take a wide variety of skills in order to be able to tackle such complex projects like this. And this is a project spanning 18 institutes and over 40 investigators to try to create these atlases. And this is only a very small subset of the people that are actually working on these projects. So I'd like to acknowledge all of them, as well as the support of the BRAIN Initiative and BICCN and BICAN for their funding and support of this project. With that, I will turn it over for you to hear more detailed elements of the projects.
Great, thank you so much, Ed. And next, I will introduce Kimberly Siletti. Kimberly Siletti is an assistant professor at the University Medical Center at Utrecht in the Netherlands. Her group focuses on the molecular mechanisms that generate cellular diversity in the brain. She completed her PhD in 2017 at the Rockefeller University with Jim Hudspeth, studying cell development in the inner ear. She followed this work as a postdoctoral researcher in Sten Linnarsson's group at the Karolinska Institute in Sweden, where she used single-cell methods to survey both developing mouse and adult human brains. Thank you so much, Kim, for being here with us today. I'll turn it over to you for your presentation.
Thank you. I'll share my screen. Okay. Yeah. So I will be talking now about some of the work to come out of the BICCN efforts that Ed already mentioned and described very nicely, and in particular, the draft version of the first human brain atlas, which was a collaboration primarily between the Allen Institute and the Karolinska Institute. And I won't spend too much time motivating this work because I know Ed has already described the importance of this work very well. This work came out of some of these earlier versions of a mouse brain atlasing efforts, where we wondered how some of this diversity was mapping to the human brain. And so we made a plan to sample the human brain. This was largely led by Rebecca Hodge at the Allen Institute. The dissection of 100 different areas from across the entire human brain. The dissections are shown here, and you can see that many of these dissections are from the cortex, given the size of the structure, but we did sample all of the other major brain regions as well. And we did this in 3 different donors, with the results being approximately 2.5 million neurons and 900,000 non-neuronal cells. And without going into too much detail, we took an iterative approach to analyzing this data. So we first pulled all the data together and clustered the data into 31 top-level superclusters, which are colored here on the t-SNE. But then we took each supercluster and analyzed it separately to produce clusters, and then took each cluster and analyzed it one more time to produce subclusters, ending up with over 3,000 subclusters. I won't go into too much detail about these different subclusters and clusters today. Obviously, there's so much to learn here. But I just want to point out a few of the major observations we made about the overall structure of the brain and the diversity that we saw. So one of the first things that we noticed about the results was that the superclusters largely reflected their developmental history. So here I'm showing a plot where each dot represents the fraction of cells to come from a specific dissection and contribute to a specific supercluster. And then these dots are colored by the region that the dissection comes from. And you can see here there's already a pattern. Many of these superclusters are region specific, for example. But you can also see that some superclusters are found across multiple regions. For example, these superclusters here are found across the cortex, hippocampus, and cerebral nuclei. And these are all from the same developmental compartment, which is called the telencephalon. And you can see the developmental history even more clearly in some migrated cells. So here I'm pointing out a supercluster that contains cells from both the thalamus and the midbrain. And indeed, there are neurons that migrate from the midbrain into the thalamus during development, suggesting that even in adulthood, they reflect this developmental history. And you can also see a developmental split in the glia. Both astrocytes and oligodendrocyte precursors split into two major groups. And these groups, again, are enriched inside and outside the telencephalon. The second major observation I want to point out is that we found extensive diversity outside the telencephalon. And this conclusion largely came from an analysis of one supercluster of neurons that we have been referring to as the splatter neurons. And that's purely because of how they looked on the t-SNE. You can see here it kind of looks like someone splashed paint on the t-SNE. And the name kind of stuck because actually it was very difficult to come up with a biological name for this grouping. This is a very diverse group of cells. It contains cells from mostly outside the cortex and the hippocampus. So here we have cells from the hypothalamus, the midbrain, and the hindbrain. It also contains over 1,100 subclusters. So this is a third of the total diversity that we found. And that was 900 more than any other supercluster in the data. So by far the most diverse supercluster of cells. It also contained excitatory and inhibitory neurons, as well as serotonergic and dopaminergic neurons, so other neurotransmitters fell into the supercluster as well. And even though we only sampled about 1,000 of these smaller groups of cells, we still ended up with 14 subclusters of each of these groups. And within these groups, we identified a lot of combinatorial expression of neurotransmitters and neuropeptides. Just as a quick example here, I'm showing the serotonergic neurons. And you can see that they express a gradient of serotonin transporter in combination with different genes necessary for excitatory and inhibitory transmission. So we wondered a lot about what this group of cells was really representing. And a student in Sten Lennerson's group, Alejandro Mossi, pointed out, so did some work profiling some of these regions using single molecule fish. And he pointed out that when he was profiling some of these neurotransmitters and neuropeptides, sometimes he would only find a neuropeptide like this one in only one cell or a few cells in the section that he was in, suggesting there are some very rare cell types in these brain regions. And I speculated that this is really what this kind of embedding might represent here. I can imagine if we're sampling neurons that might not cluster very well, they might fall, say, into the center of a large group of cells like this. So we played around with how the sampling might affect the results that we had by performing some computational experiments. We took the whole data set, and then took one supercluster, removed everything but 5 cells, reanalyzed the data, and checked where it ended up. And we found that very often the cells would end up in the splatter supercluster if they were from outside the telencephalon, so regions like the hypothalamus and the midbrain and the hindbrain. And this really suggests that these areas are very diverse to the point where if we sampled more of these cells, we would begin to see them coming out as new superclusters. And so there's a lot of diversity in these regions of the brain. I do just want to quickly point out that the cell types are still highly regionalized here. So if we take a look at this group of cells alone and plot their region of origin, we do see separations here on the t-SNE. And this really suggests, again, that if we sample these cells more deeply, as BICAN will do, that we'll start to see new superclusters come out of these cells and a much better resolution of the diversity here. So in summary, in this first draft atlas of the human brain, we noticed a lot of regional diversity. And this was especially true outside the cortex, regions like the cortex and hippocampus. And this data, as Ed already mentioned, is available to use. So for example, you can find it at the CellXGene browser, also the GitHub page from the paper. And so we encourage you to explore this data too. And I'll just end by acknowledging everyone who was involved in this work.
Great. Thank you so much, Kimberly, for a great presentation. And now I would like to introduce Rebecca Hodge. Rebecca Hodge is an assistant investigator at the Allen Institute for Brain Science, working on large-scale projects to generate comprehensive brain-wide atlases of cellular diversity in human and non-human primate brains using multi-omic and spatial transcriptomic methods. She is a member of the Seattle Alzheimer's Disease Cell Atlas Consortium, otherwise known as SEAT, where she helps to lead efforts to generate a high-resolution, multimodal cellular atlas of Alzheimer's disease. Prior to joining the Allen Institute, she completed her postdoctoral research at the University of Washington at the Center of Integrative Brain Research at Seattle Children's Research Institute, and her PhD at the University of British Columbia. Thanks so much, Rebecca, for presenting today. I'll hand it over to you.
Great, thanks very much for the introduction. I'm excited to tell you all today about the next phase of these atlasing projects that we're working on. So as Kim discussed in her talk, over the past several years, there have been significant efforts to characterize the cellular diversity of the brain across various species, including extensive research in both mice and humans using single-cell genomics techniques. But despite this remarkable progress, there really remains a need to comprehensively catalog human brain cell types across different brain regions and to understand how these cell types and the genes that they express are linked to brain function. So the Human and Mammalian Brain Atlas project that Ed introduced really aims to achieve this comprehensive characterization of cellular diversity, and we're going to do that using multi-omic single nucleus genomics assays. So these allow us to measure both the transcriptome and the epigenome of individual cells. The transcriptomic data really forms the basis for our cell type characterization and the generation of cellular taxonomies, while the epigenetic data can do things like enabling discovery of cell type specific enhancers or analysis of gene regulatory networks, for example. So for this project, we are taking a cross-species comparison approach, where we'll generate comparable atlases in human brain and in several of our closest non-human primate relatives, so macaque monkeys and marmoset monkeys. This is going to allow us to identify both conserved and species-specific features of cell types. And as Ed mentioned, cross-species data integration is really important for helping us to infer cellular properties that are difficult to measure in humans. So things such as long-range cellular connectivity, for measurements that are more tractable to perform in model species. So the sampling strategy that we're taking for this next generation of brain atlases was really designed to address some of the major challenges that Kim highlighted in her talk. We're going to be making use of multiple different dissection techniques alongside an iterative and responsive analysis plan so that we can ensure that we have comprehensive coverage of cellular diversity across all of the brain regions and all species. So specifically, we're using an atlas-guided sampling strategy for subcortical structures that have identifiable gross anatomical features in fresh frozen tissue slabs. So this involves utilizing digital atlases in combination with gross anatomical landmarks to let us achieve precise dissection of target structures. In the example that I'm showing you here, this is a rhesus macaque brain, and it has the subcortical atlas of rhesus macaque atlas parcellations overlaid onto a structural MRI that's taken from the same animal that the tissue is derived from. And that information is then matched to the structures on the tissue slab image that's shown on the left, and corresponding regions for dissection are annotated on that tissue slab image. So this basically enables us to perform accurate dissections of subcortical structures within a given tissue sample, as well as match those dissection regions across animals or across brain donors so that we can ensure that we're sampling from the same brain regions across all of the subjects included in the study. So as Kim showed you in her talk and highlighted, much of the neuronal diversity in the brain is really found in subcortical regions, and particularly in midbrain and hindbrain regions. And this diversity is complex and it's undersampled. So here, I'm just sort of reiterating this by showing you again the superclusters from the study that Kim discussed and highlighting superclusters originating from pons, medulla, and midbrain regions. And you can see that these dissections contribute heavily to the highly diverse and undersampled splatter supercluster that Kim discussed. So these regions of brain are also very anatomically complex. They contain many small nuclei that are difficult to distinguish in frozen brainstem. And you can see that I'm showing an example of a frozen human brainstem section where really there are very few reliable anatomical landmarks that we can see by eye in this type of tissue. So to address these kind of sampling challenges, in this version of the atlas, we're taking an approach to kind of exhaustively sample these brainstem structures. And this involves tiling across the entirety of the midbrain and the pons and the medulla to ensure that we're covering every structure, that no small structures are inadvertently skipped. And we also plan to very deeply sample these regions. So for example, in human, we're aiming to sample roughly 10 million total nuclei across all of the donors that are included in the study, so that we can ensure that we really capture the complete diversity of neurons in these complex brain regions. And then lastly, we're also trying to link the underlying structure of brain regions with their function. And to do that, we're pairing function localizing MRI techniques with downstream single-cell genomics and spatial transcriptomics assays. So this is largely being done in macaque animals, where there have been many years of research to develop function localizing tasks that enable researchers to characterize areas of the brain that are particularly active when an animal is presented with a specific stimulus. So the example shown here is work from our collaborators in the Freiwald lab, where they've showed that specific areas of temporal cortex are preferentially active when an animal is presented with an image of a familiar face versus a familiar object. So the macaque monkeys that we'll use in this study engage in a set of tasks. These range from things like retinotopic mapping to these tasks that are recognition of objects or faces. And the functional MRI data that results from these experiments, along with cortical parcellations from existing 3D digital reference atlases, are then mapped onto tissue slabs. And so the combination of this functional MRI and cortical parcellation data is then used to guide our dissections of cortical regions. And these will allow us to answer questions about how cellular architecture and gene expression of areas is activated in response to a certain task and how that might differ in comparison to adjacent areas that are not activated. So after roughly a year of active data generation, we've made some substantial progress. We've had a main initial focus on subcortical sampling, and we've recently achieved the milestone of completing sampling in our first human donor and plan to begin cortical sampling using these techniques I just talked about in macaque later this year. Fenna Kreenan's group, who is leading the atlasing efforts in marmoset, also recently completed full sampling of their first marmoset animal subcortex. So a key initial focus for the group has been on basal ganglia structures, where we're really aiming to create a consensus cell type atlas across human, macaque, and marmoset, as well as mouse. This is part of a larger BICAN-wide consortium effort. And we've been focusing on 8 core regions of basal ganglia. So a main goal of this work is to create atlases that use harmonized nomenclature across species to enable easy comparisons of cell types across species and to provide linkage to published work. So early results from these analyses are shown here. What you can see, basically, we're showing the regional distribution and the counts of different cellular subclasses across basal ganglia structures. And essentially, you can see that the regional distribution of these types is similar across species. However, we do see some features that may indicate species-specific specializations that require further validation. So for example, we're seeing there might be a rare or undersampled interneuron type in the human substantia nigra. So to really kind of dig into this and to be able to validate these findings, we need other methods. And one of the ways that we're going to go about doing this is by generating large-scale spatial atlases in each species. And I'm going to hand off to Stephanie, who's going to tell you about how we're doing that.
Great. Thank you so much, Rebecca, for a great presentation. And now it's my pleasure to introduce Stephanie Seeman. Stephanie Seeman is a scientist at the Allen Institute for Brain Science, working on building comprehensive spatial atlases of cell types in the human and non-human primate brain. She has also worked on projects to explore not only how different cell types in the brain are spatially arranged, but how they communicate with each other to sense the world around us. Prior to joining Allen Institute, Stephanie completed her PhD at the University of Washington. So with that, I'll hand it over to Steph.
Thanks so much. Yeah, I'm going to pick up pretty much where Rebecca left off to tell you about the spatial atlases that we're building of the human and non-human primate brains. And as you've kind of seen already, there's a lot of information to still be gained from the putting cell types back into their native anatomical context. These small brainstem nuclei are very difficult to tease apart from single cell and single nucleus approaches, as these splatter neurons have indicated. So using spatial technologies to aid in those endeavors are critical, but also there's a lot of larger questions that we can explore by looking at spatial cell type atlases, such as cell type and gene expression gradients, as well as relating those features to functional boundaries that Rebecca mentioned in the fMRI data that we will gather for particularly macaque brain samples. And so tackling these large brain samples, as Ed mentioned, is incredibly challenging. And so we need to take different approaches for these larger brains that have been traditionally used for the mouse, though we are using two similar commercially available spatial technologies, the VisGen MERScope and 10x Xenium systems, which give us imaging-based spatial transcriptomics data. And so for the relatively smaller non-human primate brains, marmoset and macaque, we can build whole brain, almost whole brain atlases in the spatial domain. And then for the larger human atlases, we'll take a more targeted approach, looking at particular regions where there's a lot of information to be gained. And so much of what we've been focused on in this initial phase of the BICAN and HMBA project is to build a robust pipeline that we can put all of these samples through to build these atlases. And so I'll tell you a bit about each of the stages of this pipeline and how we've been going about it. So the first stage is to design a gene panel. These spatial transcriptomics platforms allow us to profile upwards of 500 genes, but this is a much smaller subset of all of the genes in the genome. And so we can use the cell type taxonomies that are generated from the single nucleus data to both inform the important genes for distinguishing cell types, as well as use these taxonomies to predict our performance in recapitulating the cell types using this subset of genes. Once we've picked our 500 genes of interest, we need to prepare the tissue. These platforms have a relatively small acquisition window for the experiment, typically 1 to 2 centimeters squared, which is big enough to fit a mouse brain, but much smaller than the human and non-human primate brains that we are wanting to sample here. And so we can use MRI from the individuals as well as atlas-based templates to subdivide these brain tissues into smaller blocks that will then fit into the machines for us to do the experiments. We then take these blocks and thinly section them and pass those thin sections off to our phenomenal spatial transcriptomics team, who are running a bank of MERScopes and Xenium systems seemingly 24/7 to produce fantastic spatial transcriptomics data. When we get that data back, it goes through several processing stages. The first being cell segmentation. So we need to take images such as a DAPI image or other cell identifying stains to assign the transcripts that are measured in the spatial transcriptomics experiment to their cell of origin. And so we can use both of these pieces of information to draw boundaries around the cell nucleus and cell body to then define where the cells were and place the gene transcripts that were measured into their appropriate cell. This gives us what we call a cell by gene table, where we have cells and all of the genes that were expressed in them. Using these data and going back to the taxonomies, we can map the cell types from the single nucleus RNA-seq taxonomy to our cell types in the spatial transcriptomics data using tools that were also built at the Allen Institute and are available on our GitHub. And this is also a very circular and iterative process where we can use newly gained information about the cell types in the spatial data to update and inform the taxonomies to provide richer characterizations of the cell type identity. Since we've now cut this brain up into small pieces, we need to stitch it and put it back together. And so the final stage of this pipeline is to take all of the individual pieces of brain tissue that were run through the spatial transcriptomics experiments and stitch them back together. We've already started to see some interesting results from these data. We are also focusing first on the basal ganglia nuclei. And so there's initial information from the taxonomy suggesting that there's regional specification to where some of these cell types reside. And we indeed can see that cell types, particularly of this D1, D2 hybrid type, show remarkable patterns in their spatial context in multiple axes in the rostral-caudal axis, dorsal ventral, and also the medial lateral axes. And so this sort of richness of understanding cell types in space is, I think, a key aspect of this part of the atlasing. Similarly, we can link cell types to their connectivity and their function. Two very distinct cell types, the core and shell type of the globus pallidus internal segment, show remarkable compartmentalization in this structure that is directly linked to their input patterns from different cortical areas. And so these are just our initial findings, and we're excited to continue to look at cell types in their spatial context across these three species, not only in the basal ganglia, but brain-wide, and use this to characterize spatial domains of cell types across these species, explore conserved and divergent characteristics of these cell types, and ultimately use that to generate hypotheses about evolution and how this could be implicated in behavior and disease. And with that, I will pass it off to Elysha to tell you about tools used to view these.
Awesome. Thank you so much, Stephanie, for a great presentation. And with that, I'd love to introduce Elysha. Elysha Fiabane is a product manager on the data technology team at the Allen Institute for Brain Science. Her primary focus is on continuously improving the data visualization and analysis tools like the Cell Types Knowledge Explorer, ABC Atlas, which has been referenced throughout today's presentations, and Map My Cells, which are publicly available to neuroscientists. Previously, Elysha held product leadership positions in health technology companies like Athena Health and Pear Therapeutics. Elysha holds a bachelor's of arts in linguistics and sociology from Boston College. Thanks so much, Elysha, for being here with us today, and I'll hand it over to you.
Excellent. Thank you so much. And thank you so much to the previous speakers for showing us how all this data is collected. Unlike them, I'm not a scientist. My role at the Allen Institute is to make all of that data and analysis available to the public and make it more useful to other neuroscientists. So we've created what we call the Brain Knowledge Platform here at the Allen Institute. It really is designed to connect data across species, modalities, anatomical space, and developmental time, and create a set of tools that allow users to engage with the data and discover new insights. I'm going to talk today specifically about the ABC Atlas because it is where you can go to interact with all of the data that you heard discussed today. You'll see the Siletti data there at the ABC Atlas. You can filter and color by all of the various classes, subclasses that were described, as well as human data and as well as several whole mouse brain data sets and the CAD Alzheimer's disease data. So what is the ABC Atlas? It is a place where you can explore and compare whole brain data. It's flexible, it's data-driven, it has a scalable infrastructure to support new and expanding science worldwide. It also allows you to compare data sets side by side. There's lots of tools out there that are designed to showcase one specific data set or one specific set of research. For our purposes, because we are producing so much data across species and for so many different projects, we wanted one place that we could bring it all together. We also wanted a place where we could connect the science and the scientists working on these projects. So the platform encourages collaboration. It includes the ability to share interactive deep links to cite any of the research and tools that you interact with there, and also to download images. It's also connected deeply to the raw data sets. So you can download that data and analyze them in your own way. How do you get there? You can search for ABC Atlas. It should get you there. But this is a specific link to give you an overview of all the data that's available. And just click on that first blue link to get to the ABC Atlas. So what's it good for? What does it do? The questions that ABC Atlas is good at answering, for example, are genes X and Y co-expressed in the same cell type? You can filter and color by gene expression. Where is cell type X in the brain? We have the spatial transcriptomic data that actually shows you that spatial distribution. Some questions that ABC Atlas isn't great at yet. So it does not show the morphology of the cells, and it does not show the synaptic connections. So we have other tools that let you explore that. Cell type knowledge explorer is good for exploring that, and it doesn't show you in quite the same way. Again, is the soma cell type X larger than some of cell type Y? That type of morphological data is not shown. We have lots of plans to improve the product. So if you have suggestions for things you'd love to see, things you want to do with it, please let us know. So I'll give you a quick tour here. Please, I encourage you to log in and play with it yourself. A couple of highlights of what it can do. As you're exploring the data sets, you can use our tooltips to interrogate any of the cells. So each of the dots here that you see is a cell. You can understand what cell type it is, depending on what you're coloring by. It also shows the common coordinate framework overlaid, so you can see the anatomical region that it's a part of. You can also look at gene expression data. So you can filter and color by all the cell properties, cell class, as well as other cell properties and features that are available in the data set. This is the same data that's available for download. So we're just taking it and making it accessible through this visualization tool. we also have the ability to compare views. So you can see the same data set and highlight different aspects of it and compare. So for example, if you're looking for at the same spatial location, and you can color by the cell type as well as a particular gene expression and see how it compares. You can also compare different data sets side by side as well. We also allow the sharing and citation tool, so you can share these insights that you're generating with your colleagues. Just a few notes on support resources. If you're in there and you want some help, we have this great new resource. The link is right here that will give you resources to help you navigate and answer some common questions. And with that, I think I'm getting, I've overstayed my welcome in terms of the timing. We have to get going. Yes, what's next? We want to make it more accessible with generative AI. And also we have a lot of community outreach initiatives that we're doing with both the scientific and non-scientific communities. We also want to link it to our ecosystem of tools. All of the developers out there can build on top of the data and tools that we provide. And then, as you saw, we have a lot of data coming through the pipeline, and we are excited to bring all of that to you in the ABC Atlas. And so with that, I will hand it back to Jimena.
Thank you. Thank you so much, Elysha, and thank you to all the panelists. So now I realize I didn't introduce myself, team. So thank you everyone for being here. I'm a program manager of inclusive research at Allen Institute and part of this team as well. So now we're going to transition to the Q&A. So thank you to those who have asked questions throughout the webinar. Please review the answers provided by the panelists in the Q&A tab at the bottom of this webinar. We are also going to provide, as I mentioned earlier, a Q&A fact sheet when we send out the thank you to you all for being here today. So now if all panelists can please go back on camera, I'm going to ask some questions that were sent to us via the registration forms. So first, I'll start with this question, and this one is for Stephanie or Rebecca. What is the most striking difference and most obvious similarities between human and non-human primate atlases?
I don't know, Steph, if you want to take that one, but I would say my opinion is that it's a little early in our comparisons to really have a great answer to that. I think we are just currently generating a lot of data for the projects, and I think in several years, when we've generated more comprehensive data sets across all these different species, we'll be able to have a better answer for that. But I'll also let Steph weigh in.
Yeah, I think that's definitely true. It's still a little early, though. I think some initial data from the spatial experiments across the 3 species has shown that for some cell types, there's potentially quite variable proportions or spatial patterns of those cell types across the species. But then other cell types, such as those core and shell cell types in the globus pallidus, seem to be showing some conservation even down to the mouse. And so I think it will be a sort of cell type specific and dependent answer that will be interesting to uncover.
Awesome, thank you.
To that question? So I think the findings are just coming from these newer atlases, but we've been doing comparative studies for some time that have revealed some pretty interesting things. The first thing is that the overall cellular organization and diversity is quite similar between a mouse and a human, despite a 1,000 times increase in the size. And so that was a bit of a surprise. I think we might have expected there to be much greater diversity. And many of the same sort of principles of where the diversity is, such as deep in the brain, are true across mammals. And so you see this in mouse as well as human, for example. Down to a certain level, things are conserved, but then when you get to a finer granularity, many species differences start to emerge. You can see elaborations of certain kinds of cells that relate to the functional architecture. There are many, many differences in gene expression. And then when you get to our closest relatives, like chimpanzees and gorillas, really the cellular architecture doesn't change very much, but there nevertheless are gene differences across species. And these seem to point to differences in how neurons connect with one another and make functional networks. So things associated with synapses and neuronal connectivity. So, these are just a few hints of the things that you can see, but principles begin to emerge pretty quickly. But at some point it becomes empirical. You really have to look at the comparison. And so these resources are a key for being able to look at those things for any particular question of interest.
Awesome. Thank you so much. And a new question just came in through the chat. So this is for anyone who would like to answer. What is the best tool that you guys have to visualize and quantitatively measure neuron density, size, and morphology across different cortical layers in different brain regions?
I could try and take a stab at that. I'm not sure if they mean tools, public tools of data that's already generated, or the tools that we use internally to get that data. I think those are a lot of different quantitative pieces of information that don't all come from one approach. Some of the work that wasn't addressed here, but is also part of the HMBA, are patch-seq experiments, which do in vitro single-cell electrophysiology and can fill those cells with a dye that then allows for detailed reconstruction of the morphology, and doing that in a principled way across layers and areas has shown nice data about neuron size and morphology in those different regions. And then I think neuron density is likely to come from some combination of the spatial and single nucleus RNA-seq data, where we can really profile many thousands of millions of more cells in those assays compared to something like a patch-seq experiment, which is a very low throughput but highly detailed approach.
Great. Thank you so much, Stephanie, for that response. Would anyone else like to add to that, or should we move on to the next question? No additions? Okay, awesome. So the next question is, if everyone could give a bold prediction, what kind of technology do we still need to profile the whole brain molecular omics? I mean, the whole brain, and when? And if we can start from Ed.
Very specific. Okay. Well, the biggest problem is scale. So, we have some techniques here which are capable of looking at very, very high resolution with multiplexed assays, but they tend to be small format. And so the biggest problem, I think, with doing this is a scale up of technologies to let you do an omics level readout on a brain section scale. And there are actually a number of technologies that are being developed along these lines. One of them is called StereoSeq, which is a sequencing-based spatial assay that can be run on a chip that can be almost the size of a human hemisphere. And so that is pretty early days with this. It's already been used and published on a macaque brain. And we've been working with this as well now. But the larger format versions of this can actually be applied to maybe half hemispheres of a human brain. So the biggest challenge there is just the cost. They're extremely expensive to apply. But one can begin to do this sort of thing with a true omics kind of readout. Now, on the other hand, you might want to get actual volumetric information about the brain rather than thin sections and get a more complete understanding of that. And there, the methods are being developed to do large format, cleared tissue, volumetric imaging of some set of proteins and mRNA molecules. This has been commercialized through a company called Life Canvas. Many of the techniques are developed by Kwang-Hoon Chung at MIT. And so there you get, less of a multiplex, but you can actually plausibly do a whole human hemisphere as a volume. And so I think these techniques are quickly becoming possible, if extremely expensive. And I think the human use case is the thing that's really driving it. The brain is enormous. You have to look over large areas to really understand what's happening in a disease, for example. And so there's a serious need for these sorts of things for neuropathology and understanding of disease. But they're still pretty nascent at this point.
Thank you so much, Ed, for that response. We also have a really great question that came in from the registration forms, and this question is for Rebecca. So are there any plans for expanding the data set to account for individual variability in the human population?
Yeah, so I didn't get a chance to talk about this today, but there is a population component to the study that we designed. So the whole brain atlasing efforts really by necessity are small number of donors, but we are profiling two particular areas across larger populations in human, macaque, and marmoset to take advantage of sort of the benefits of each of those species and the questions that we can answer using the metadata that we have associated with those species. So in human, we're interested in understanding how do cell types and gene expression change across things like age or sex in a diverse cohort. In macaques, we're actually performing this study using macaque monkeys that are from Cayo Santiago in Puerto Rico. So these are sort of a naturalistic colony of macaques that are periodically culled to maintain population, and they have a huge number of different social metrics that are recorded over their lifespan, so we can understand, how those vary across this population of monkeys. And in marmosets, this is work that's being done by Guoping Feng's lab at MIT, but they're actually, lab-based animals that can perform particular tasks related to the areas that we're sampling in. And so we're sampling subgenual cingulate cortex and medial entorhinal cortex across populations of 50 to 80 donors in each of those species. So yeah, that is also part of the study.
Thank you so much, Rebecca, for that response. And I think we have maybe time for just one last comment. So I don't know if anyone on the panel would like to share one last comment as a takeaway for those who joined us today.
I'll take the opportunity. I think that, we really would like to convey the results of this project as it's done, so that the community is aware of it and can begin to take advantage of it. The goal is to create a community resource that will accelerate everyone's research in the field. And so that's really part of the intention of doing this and of communicating both the state of the science and the state of the tools. And we really welcome your feedback to try to make this more impactful and more useful for your research so that it achieves this goal.
Thank you so much, Ed, for that. And Elysha, can you share where individuals can share feedback or questions as they're exploring our data tools?
Yes, absolutely. If you go to our community forum, I can share a link to that, but we have a forum that is monitored by the scientists and the tool creators, and if you ask questions there, we'll jump in and help you out. So I really, really appreciate all the feedback that folks have given already, and we find it super valuable.
Awesome. Thank you so much. So everyone who joined today, thank you so much. We are going to provide the links to all of the papers presented and data that was presented today, as well as links to the resources that Elysha is mentioning. After this webinar, a survey will pop up. If you attended, please respond to that survey. Thank you so much, everyone, for joining, and have a great day. Thank you.